Hello everyone, welcome to my channel. My name is Urman and today we are going to talk about the big ideas from ARKInvest from 2021, and this episode is actually the last episode because we will be talking about part 15, Cell and Gene Therapy Generation 2. Well, this part outlines that the second generation of cell and gene therapies could shift from liquid to solid tumors, autologous to allogeneic cell therapies, and from ex vivo to in vivo gene editing. ARK estimates that the new cell and gene therapy innovations could increase the total addressable market for oncology therapeutics by more than 20-fold. Your first question might be, what is gene editing? Well, one of these techniques is called CRISPR, and Jennifer Doudna is gonna explain this right now to you.
What I want to do now is I want to tell you what the CRISPR technology is, what it can do, where we are today, and why I think we need to take a prudent path forward in the way that we employ this technology. When viruses infect a cell, they inject their DNA. And in a bacterium, the CRISPR system allows that DNA to be plucked out of the virus and inserted in little bits into the chromosome, the DNA of the bacterium. And these integrated bits of viral DNA get inserted at a site called CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. A big mouthful. You can see why we use the acronym CRISPR. It's a mechanism that allows cells to record over time the viruses that they have been exposed to. And importantly, those bits of DNA are passed on to the cell's progeny. So cells are protected from viruses not only in one generation, but over many generations of cells. This allows the cells to keep a record of infection, and as my colleague Blake Wiedenheft likes to say, the CRISPR locus is effectively a genetic vaccination card in cells. Once those bits of DNA have been inserted into the bacterial chromosome, the cell then makes a little copy of a molecule called RNA, which is orange in this picture, that is an exact replicate of the viral DNA. RNA is a chemical cousin of DNA, and it allows interaction with DNA molecules that have a matching sequence. So those little bits of RNA from the CRISPR locus they bind to protein called Cas9, which is white in the picture, and form a complex that functions like a sentinel in the cell. It searches through all of the DNA in the cell to find sites that match the sequences in the bound RNAs. And when those sites are found, as you can see here, the blue molecule is DNA. This complex associates with that DNA and allows the Cas9 cleaver to cut up the viral DNA. Makes a very precise break. So we can think of the Cas9 RNA sentinel complex like a pair of scissors that can cut DNA. It makes a double-stranded break in the DNA helix. And importantly, this complex is programmable. So it can be programmed to recognize particular DNA sequences and make a break in the DNA at that site. As I'm going to tell you now, we recognized that that activity could be harnessed for genome engineering to allow cells to make a very precise change to the DNA at the site where this break was introduced. That's sort of analogous to the way we use a word processing program to fix a typo in a document. The reason we envisioned using the CRISPR system for genome engineering is because cells have the ability to detect broken DNA and repair it. So when a plant or animal cell detects a double-stranded break in its DNA, it can fix that break either by pasting together the ends of the broken DNA with a little tiny change in the sequence at that position, or it can repair the break by integrating a new piece of DNA at the site of the cut. So if we have a way to introduce double-stranded breaks into DNA at precise places, we can trigger cells to repair those breaks by either the disruption or incorporation of new genetic information. So if we were able to program the CRISPR technology to make a break in DNA at the position at or near a mutation causing cystic fibrosis, for example, we could trigger cells to repair that mutation. Genome engineering is actually not new. It's been in development since the 1970s. We've had technologies for sequencing DNA, for copying DNA, and even for manipulating DNA. And these technologies were very promising, but the problem was that they were either inefficient or they were difficult enough to use that most scientists had not adopted them for use in their own laboratories, or certainly for many clinical applications. So the opportunity to take a technology like CRISPR and utilize it has appeal because of its relative simplicity. We can think of older genome engineering technologies as similar to having to rewire your computer each time you want to run a new piece of software. Whereas the CRISPR technology is like software for the genome. We can program it easily using these little bits of RNA. So once a double-stranded break is made in DNA, we can induce repair and thereby potentially achieve astounding things, like being able to correct mutations that cause sickle cell anemia or cause Huntington's disease. I actually think that the first applications of the CRISPR technology are going to happen in the blood, where it's relatively easier to deliver this tool into cells compared to solid tissues.
Cancer therapies are shifting from liquid to solid tumors. Typically, cancer therapies are tested on liquid tumors first, because solid tumors, however, they comprise of 88% of diagnosed cancers. And the US Food and Drug Administration, also known as the FDA, they approved Gleevec, which is an oral chemotherapy, after 10 years of trials, and 7 years of which were in solid tumors, which is also represented in the graph on the right. It's called the cancer therapy timeline. On the x-axis we have time. The green color represents the liquid tumor and the red color represents the solid tumor. And indeed, we started with Gleevec in 1998. The therapy got approved for liquid tumors in 2001. Then we moved to the solid tumor for the Gleevec in 2001. The therapy got approved in 2008. And this timeline suggests now that the FDA could approve the first CAR-T therapy for solid tumors in 2025. And because of artificial intelligence, gene editing, and next-generation sequencing, the failure rates and time to market, they should fall, accelerating approval rates. So what are examples and consequences of CRISPR gene editing?
Right now, a lot of the work that's going on applies to animal models of human disease, such as mice. The technology is being used to make very precise changes that allow us to study the way that these changes in the cell's DNA affect either a tissue or, in this case, an entire organism. Now, in this example, the CRISPR technology was used to disrupt a gene by making a tiny change in the DNA in a gene that is responsible for the black coat color of these mice. Imagine that these white mice differ from their pigmented littermates by just a tiny change at one gene in the entire genome. And they're otherwise completely normal. And when we sequence the DNA from these animals, we find that the change in the DNA has occurred at exactly the place where we induced it using the CRISPR technology. Additional experiments are going on in other animals that are useful for creating models for human disease, such as monkeys. And here we find that we can use these systems to test the application of this technology in particular tissues, for example, figuring out how to deliver the CRISPR tool into cells. We also want to understand better how to control the way that DNA is repaired after it's cut, and also to figure out how to control and limit any kind of off-target or unintended effects of using the technology. I think that we will see clinical application of this technology, certainly in adults, within the next 10 years. I think that it's likely that we will see clinical trials and possibly even approved therapies within that time, which is a very exciting thing to think about. And because of the excitement around this technology, there's a lot of interest in startup companies that have been founded to commercialize the CRISPR technology, and lots of venture capitalists that have been investing in these companies. But we have to also consider that the CRISPR technology can be used for things like enhancement. Imagine that we could try to engineer humans that have enhanced properties, such as stronger bones or less susceptibility to cardiovascular disease, or even to have properties that we would consider maybe to be desirable, like a different eye color or to be taller, things like that. Designer humans, if you will. Right now, the genetic information to understand what types of genes would give rise to these traits are mostly not known. But it's important to know that the CRISPR technology gives us a tool to make such changes once that knowledge becomes available. This raises a number of ethical questions that we have to carefully consider. And this is why I and my colleagues have called for a global pause in any clinical application of the CRISPR technology in human embryos to give us time to really consider all of the various implications of doing so. And actually, there's an important precedent for such a pause from the 1970s, when scientists got together to call for a moratorium on the use of molecular cloning until the safety of that technology could be tested carefully and validated. So, genome-engineered humans are not with us yet, but this is no longer science fiction. Genome-engineered animals and plants are happening right now. And this puts in front of all of us a huge responsibility to consider carefully both the unintended consequences as well as the intended impacts of a scientific breakthrough.
Oncology trials are shifting from autologous to allogeneic cell therapies. So what is the difference? Allogeneic cell therapy involves cells off the shelf or donated, whereas autologous cell therapy modifies the patient's own cells. And one risk of allogeneic therapies is graft versus host disease, causing a patient's immune system to attack the newly engineered cells. But despite this risk, the allogeneic cells can facilitate therapies at earlier stages of cancer and reduce costs by an order of magnitude. So on the right, they put this in a graph. The dark purple color represents the autologous one, and the light purple color is allogeneic. But if we focus on autologous, the cost is much higher, so more than $100,000, but there is no risk of cell rejection. On the other hand, we have the light purple color, allogeneic. The cost is much lower, so less than $10,000. It's scalable. There is product consistency. We have an ability to redose, and there's expanded access to the sickest patients. And on the right, we have the autologous and allogeneic trials composed of recruiting, completed, and approved therapies. And among these gene therapy trials today, 40% of those completed and 47% in the patient's recruitment phase, they were allogeneic. And in ARK's view, the shift to allogeneic trials is something that will continue in the future.
So we are studying the convergence of sequencing and artificial intelligence and CRISPR gene editing. We're seeing all kinds of platforms converge here, but this is probably one of the most important set of convergences. Is it wild-eyed to be thinking that as this convergence evolves and we get deeper and deeper into understanding what's going on, that we could cure most diseases? Is that outrageous to think? Or I'm just dreaming, I realize it's not 50, it's 15, 20 maybe, a long time away, but is it a crazy thought?
Well, I would, it's not a crazy thought, but I would modify it a little bit, just to be clear. So I think that if we start imagining what happens in the future, in the coming years and decades, I think it's not unreasonable to imagine that genome editing will allow diseases that are caused by a single gene to be corrected and cured. So that is just, it's kind of mind-blowing to think about it. And it really does allow a very clear path to be envisioned, at least, for personal medicine, because this is really about fine tuning. You can take this tool and you can literally program it to make the targeted change that one wants. And so that can be tailored to individuals based on their DNA sequence. So that's where the DNA sequencing obviously comes in. Right. But that being said, of course, there are many diseases and conditions that result from multiple genes that are the way they are, and then of course lots of diseases like COVID-19 comes to mind that are not caused by genetics necessarily, but are caused by some external infectious agent. Although it's possible that that one could use genome editing there to help boost up the immune system of affected individuals. So I think that there's really lots of opportunities, but my guess is that initially the targets for genome editing in biomedicine will be focused on monogenic diseases, because those are the ones where it's easiest to envision how this technology will address those.
Gene therapies could shift from ex vivo to in vivo editing. Regarding the differences, ex vivo modifies a patient's cells outside the body and then transplants them back into the patient, whereas in vivo gene therapy modifies a patient's cells inside the body. The dark purple color represents ex vivo and the light purple color represents in vivo. If we focus on ex vivo, the cost is significantly higher and we have the ability to check for edited cells before the transduction. But if we focus on in vivo, the light purple color, the cost is much lower. We do not have the ability to check for edited cells before the transduction, but it allows for product consistency, non-toxic conditioning regimens, and we have expanded access to treatable diseases such as muscle disorder. On the right, ARK also puts here the clinical trials with active genome editors. Well, until 2015, we only had 7 of them that were ex vivo, and from 2016 to 2020, this grew to 34 ex vivo, and in vivo grew from 0 to 8. And unlike ex vivo, in vivo therapies cannot check edited cells before the transduction. But that said, in vivo gene therapy is more effective and easier to manufacture and scale, and it enables more access to the liver, eye, central nervous system, and muscles.
You see kind of 3 stages to gene editing. We're in the first stage right now. Can you give us a sense of where we are right now and where we are going from here?
Of course. So the first stage, I like to call it CRISPR-Cas9, and we think of them, as stages, but also that they can all work together. They're all just going to be really important for different disease indications. So the first one, CRISPR-Cas9, you can think of it like molecular scissors. And so we cut the DNA on both strands, and then we allow the body to basically repair those strands and, get those ends joined back together. And you can think about it like a white piece of paper. And so if you cut that piece of paper and then you put it back together again, well, it's not going to be the same as it was before you did that, right? So, this could be really good, though, for a few different type of disease indications. And so one could be ATTR amyloidosis, which is when you create too much of a certain protein, the amyloid protein, namely in this condition. And Intellia is working on this. And so you disrupt the gene because you need to create less protein. Another example would be Crisprtherapeutics, and they're working on sickle cell and beta thalassemia, which are blood diseases. And essentially what happens there is you want to sort of disrupt one gene to reactivate another gene. And so that's been seen to be really helpful there. And then we can go into sort of some of the more innovative or different types, which just basically increase the functionality. And so one example is, let's say you think of base editing. There's one company working on that there that is IPO'd. It's called Beam. And what essentially it is you can think about it like a pencil and an eraser. And so if you think about it, you're on that white piece of paper again. In the pencil and eraser, you might have some lead, little debris. You might have some little, eraser debris, but essentially your paper will look a lot better than your first paper would have. And so this isn't going to work for everything, though. It will work for 1/3 of all known genetic disease mutations because it needs to correct the base pairs that I just heard the last caller speaking about as well. And so this could be really exciting. And some companies working on Beam, like I mentioned, they're working on it for sickle cell, beta thalassemia, and Verve is working on it for the PCSK9 gene to actually target LDL or bad cholesterol.
Okay, so if it's good for a third of diseases, you also cover stem cell immunotherapy technologies as well. What's at the forefront for those two areas?
Sure. So immunotherapy, a lot of really interesting things going on. And actually gene editing plays a big part in there as well. So, we have things like CAR T cell therapy, which is basically like you can take cells out of your own body, have them genetically engineered to be cancer killing, and then be put directly back into your body. You can use your own cells, which would be called autologous, or you can use someone else's cells, donor cells, that would be already ready to go on the shelf when you get there, and that would be called allogeneic. So these innovations are happening both with CRISPR gene editing and as well as, through other mechanisms as well.
Gene therapy and editing trials have increased fivefold since 2010. ARK puts this in a graph called the gene therapies stages of advancement. On the x-axis we have on the left approved drugs, which is the red color. In the middle we have active trials, which is the green color. And on the right we have expected commercializations, which is the purple color. And to date, the FDA has approved only 10 gene therapies, but as of year-end 2020, 712 active gene therapy clinical trials were on the way, and 238 of them initiated in 2020. And adjusting for the typical trial fail rate, roughly 170 gene therapies are likely to be approved and commercialized during the next decade. One form of gene editing with increased functionality is called base editing.
My laboratory recently succeeded in developing such a capability, which we call base editing. The story of how we developed base editing actually begins 3 billion years ago. We think of bacteria as sources of infection, but bacteria themselves are also prone to being infected, in particular by viruses. So about 3 billion years ago, bacteria evolved a defense mechanism to fight viral infection. That defense mechanism is now better known as CRISPR. And the warhead in CRISPR is this purple protein that acts like molecular scissors to cut DNA, breaking the double helix into 2 pieces. If CRISPR couldn't distinguish between bacterial and viral DNA, it wouldn't be a very useful defense system. But the most amazing feature of CRISPR is that the scissors can be programmed to search for, bind to, and cut only a specific DNA sequence. So when a bacterium encounters a virus for the first time, it can store a small snippet of that virus's DNA for use as a program to direct the CRISPR scissors to cut that viral DNA sequence during a future infection. Cutting a virus's DNA messes up the function of the cut viral gene and therefore disrupts the virus's life cycle. Remarkable researchers, including Emmanuel Charpentier, George Church, Jennifer Doudna, and Fun Zhan, showed 6 years ago how CRISPR scissors could be programmed to cut DNA sequences of our choosing, including sequences in your genome, instead of the viral DNA sequences chosen by bacteria. But the outcomes are actually similar. Cutting a DNA sequence in your genome also disrupts the function of the cut gene, typically, by causing the insertion and deletion of random mixtures of DNA letters at the cut site. Now, disrupting genes can be very useful for some applications. But for most point mutations that cause genetic diseases, simply cutting the already mutated gene won't benefit patients because the function of the mutated gene needs to be restored, not further disrupted. So cutting this already mutated hemoglobin gene that causes sickle cell anemia won't restore the ability of patients to make healthy red blood cells. And while we can sometimes introduce new DNA sequences into cells to replace the DNA sequences surrounding a cut site, that process unfortunately doesn't work in most types of cells, and the disrupted gene outcomes still predominate. Like many scientists, I've dreamed of a future in which we might be able to treat or maybe even cure human genetic diseases. But I saw the lack of a way to fix point mutations, which cause most human genetic diseases, as a major problem standing in the way. Being a chemist, I began working with my students to develop ways on performing chemistry directly on an individual DNA base to truly fix, rather than disrupt, the mutations that cause genetic diseases. The results of our efforts are molecular machines called base editors. Base editors use the programmable searching mechanism of CRISPR scissors, but instead of cutting the DNA, they directly convert one base to another base without disrupting the rest of the gene. So if you think of naturally occurring CRISPR proteins as molecular scissors, you can think of base editors as pencils, capable of directly rewriting one DNA letter into another by actually rearranging the atoms of one DNA base to instead become a different base. Now, base editors don't exist in nature. In fact, we engineered the first base editor, shown here, from 3 separate proteins that don't even come from the same organism. We started by taking CRISPR scissors and disabling the ability to cut DNA, while retaining its ability to search for and bind a target DNA sequence in a programmed manner. To those disabled CRISPR scissors, shown in blue, we attached a second protein in red, which performs a chemical reaction on the DNA base C, converting it into a base that behaves like T. Third, we had to attach to the first 2 proteins the protein shown in purple, which protects the edited base from being removed by the cell. The net result is an engineered 3-part protein that for the first time allows us to convert Cs into Ts at specified locations in the genome. But even at this point, our work was only half done, because in order to be stable in cells, the 2 strands of a DNA double helix have to form base pairs. And because C only pairs with G and T only pairs with A, simply changing a C to a T on one DNA strand creates a mismatch, a disagreement between the 2 DNA strands that the cell has to resolve by deciding which strand to replace. We realized that we could further engineer this 3-part protein to flag the non-edited strand as the one to be replaced by nicking that strand. This little nick tricks the cell into replacing the non-edited G with an A as it remakes the nicked strand, thereby completing the conversion of what used to be a CG base pair into a stable TA base pair. After several years of hard work led by a former postdoc in the lab, Alexis Comor, we succeeded in developing this first class of base editor, which converts Cs into Ts and Gs into As at targeted positions of our choosing. Among the more than 35,000 known disease-associated point mutations, the 2 kinds of mutations that this first base editor can reverse collectively account for about 14%, or 5,000 or so, pathogenic point mutations. But correcting the largest fraction of disease-causing point mutations would require developing a second class of base editor, one that could convert A's into G's or T's into C's. Led by Nicole Gadelli, a former postdoc in the lab, we set out to develop this second class of base editor, which in theory could correct up to almost half of pathogenic point mutations, including that mutation that causes the rapid aging disease progeria. We realized that we could borrow, once again, the targeting mechanism of CRISPR scissors to bring the new base editor to the right site in a genome. But we quickly encountered an incredible problem. Namely, there is no protein that's known to convert A into G or T into C in DNA. Faced with such a serious stumbling block, most students would probably look for another project, if not another research advisor. But Nicole agreed to proceed with a plan that seemed wildly ambitious at the time. Given the absence of a naturally occurring protein that performs the necessary chemistry, we decided we would evolve our own protein in the laboratory to convert A into a base that behaves like G, starting from a protein that performs related chemistry on RNA. We set up a Darwinian survival of the fittest selection system that explored tens of millions of protein variants and only allowed those rare variants that could perform the necessary chemistry to survive. We ended up with a protein shown here, the first that can convert A in DNA into a base that resembles G. And when we attach that protein to the disabled CRISPR scissors, shown in blue, we produced the second base editor, which converts A's into G's and then uses the same strand nicking strategy that we used in the first base editor to trick the cell into replacing the non-edited T with a C as it remakes that nicked strand, thereby completing the conversion of an AT base pair to a GC base pair.
To conclude, sizing the opportunity, ARK estimates that allogeneic cells and cellular immunotherapies could create $250 billion in incremental revenues, and they put this in a fancy graph on the right called the liquid and solid tumor therapies, the total addressable market. And the x-axis represents stage, moving from late stage on the left to the early stage on the right, and the y-axis represents the tumor, moving from liquid tumor on the bottom to solid tumors on the top. And innovation in cellular immunotherapies, including TILS, TCRs, and CAR T-cell therapies, could increase the total addressable market by nearly threefold, adding $30 billion to the current $13 billion total addressable market. And allogeneic therapies enable easier applications in earlier stages of cancer, potentially increasing the total addressable market by $70 billion. The combination of cell therapy innovation and allogeneic cells could add an additional $150 billion and increase the overall total addressable market for oncology gene therapy by roughly 20-fold to more than $260 billion. And in vivo gene therapy could enable gene editing to cure thousands of rare diseases over time. Thank you everyone for watching this video. I really hope you enjoyed it and that you actually learned something. It was the last video of this series, so thank you very much.